Hello, and welcome to this session on how to write a science investigation report. Although I will focus on biology-specific examples, most of the content of this lesson will be applicable to all sciences. So our aim for this session is to understand the basic sections of a general science investigation write-up. And by the end of this session, you should be able to list the sections and their orders within a report and apply this information to a science experiment and use it in order to write a full investigation report yourself. Now, first, I'm just going to introduce you to the 9 main sections of a science investigation report. So we always follow the general structure of title, then aim, hypothesis, variables, description of the method, results table and graph, conclusion, and finally evaluation. So the plan for this session is we're going to go through each of these sections individually, and in order to illustrate and give you examples of how to write each section, I'm going to use the experiment below as my example. So you can see in this experiment we have 4 plants set up in containers with soil, and each plant has been placed at a different temperature. In this experiment, the height of the plant is measured before and after being placed at each temperature in order to see if the temperature affects the plant's growth. So I'm going to keep coming back to this experiment as we move through each of the sections. So firstly, we're going to look at what should hopefully be the simplest section, and this is the title. My first tip for this is you shouldn't use a generic title like biology investigation or plant experiment. Your title should instead briefly summarise what the report is about for your reader, and it should not be identical to the aim, which we're going to come back onto next. So a possible example of an acceptable title for this experiment would be Plant Growth and Temperature, or Temperature and Plant Height. There's some other ones that would be fine as well. Both of these are short and snappy, but they're detailed enough that I know when I read them what this person is investigating in their experiment. Our next section in an investigation report after the title will always be the aim. The aim details what you're trying to find out by doing an experiment or investigation. However, before you start to write their aim, there is a key step that a lot of people often miss. You must first identify the independent and dependent variables for the experiment so that they can go into your aim. Now let's break them down first of all and explain what these terms mean. So firstly, variables. So a variable is a factor that can be changed in an experiment, any factor. So the independent variable is the factor that is going to be changed in your experiment. Remember, you should only ever be changing one factor at a time. Most experiments in school, you will only focus on changing a single factor, so the variable you're changing is the independent variable. Now, the dependent variable is different. This is the factor that is measured in the experiment. So this is the factor that you'd be writing down in your results table as you went through your experiment and gathered your results. A lot of people get easily confused between the independent and dependent variables, and it's really essential that you understand which is which and that you're able to pick them out in experiments. It's not only important for investigation write-ups and for the assignment, it's also important in the exam. So one way I suggest to remember is to always use this mnemonic, I see danger mouse. So you'll notice I have the old cartoon character up on the top right of my screen. And whenever you think of independent or dependent variables, try and train yourself to imagine this character. Then you can think, I see danger mouse. So I see DM. So I see danger mouse. IC stands for independent variable is change, and DM stands for dependent variable is measured. So this is a nice simple way to remember which one is which. And now we're going to try and put those into practice. So in our experiment that we're focusing on as the example today, the variable of the factor that was altered or changed in the experiment was temperature, and the plants were set up at different temperatures to do that. So to figure out the independent variable, you're looking for the thing they changed in the experiment, and for this experiment it was temperature. The dependent variable, the factor they measured, I mentioned at the start, was the change in the height of the plant, because they measured the plant at the start of the experiment and they measured it at the end of the experiment to see if it increased in height and by how much. So now we know both of our variables, our independent being temperature and our dependent being change in the height of the plant, then we can finally create our aim. Now, aim should always have the following structure. They always start with to investigate or to find out the effect of, and then you put in your variables after that. You just slot in your independent and your dependent. And this is why it's really, really important that you work those out first. So in our example, we're going to say to investigate the effect of temperature, which is our independent variable, on the height of plants, which is our dependent variable. So the aim in total is to investigate the effect of temperature on the change in height of plants. So now we're going to move on to our third section, which is the hypothesis. The hypothesis is where you predict what you think is going to happen in your experiment with a justification of why. An important point here is that this doesn't need to be correct. It should be an educated guess based on what you know already and the background information you have on that topic through your lessons. So for the hypothesis, you should try and use the structure, I think, then whatever you do think is going to happen, and then because, and then give your justification. Another tip is that your hypothesis needs to be related to your aim. So again, try and mention your independent and dependent variables here. So an example for our experiment would be, I think that as the temperature increases, the overall height of the plants will increase because plants need warmer temperatures to survive. So in that, I mentioned what I thought was going to happen in the experiment. I mentioned both my independent and my dependent variables, and I also mentioned my reason, my justification for why I thought that would happen. So next we're going to move on and look at variables. Now we've actually already done a lot of the work here already because we selected our independent and dependent variables earlier to make our aim. So in this section, you list the independent and dependent variables along with the constant or controlled variables in your experiment. As we know from earlier, the independent variable is the variable that was changed in the experiment and the dependent was the variable that was measured. Constant variables instead are factors that were kept the same or constant during the experiment. So essentially, there are many potential variables in an experiment, lots of factors, that could have an effect on your results. We only change one variable or factor, the independent variable, and we hope that it will have an effect on the dependent variable which we are measuring. Now, essentially, as a good scientist, you want to try and ensure wherever possible that you've controlled or kept any other factors constant so that they don't accidentally affect your dependent variable and therefore your results. So you keep them constant. And it depends on your experiment what ones are there, which variables we try and keep the same. Some examples of possibilities would be keeping the concentrations or volumes of solutions or liquids the same, or maybe keeping the mass of solids the same, or controlling the temperature or pH of solutions, or even how long you leave the experiment to run for. There's lots and lots of different examples of variables that you could list here, and for your own experiment write-up, you should aim to list as many as possible. And I would always say you should aim for at least a minimum of 3. So let's look at an example of the variable section. So one final tip is to ensure you write variables as your heading for this section, and then you clearly indicate which variables come under which type, like in the following example. So for our experiment, we already discussed that the independent variable is temperature, and the dependent would be the change in the height of the plant. In here, I've also added in brackets centimetres, because that's what I'd measure the height of the plant in. I wouldn't put this in the aim, but I like to include my units here, and I've done the same thing for temperature, so I've put degrees Celsius. For the constant variables, I would look at this experiment and think, what factors would I need to keep the same? I'm only changing temperature and I'm measuring the height of the plant, so what other things could have an effect on my results? So some examples here would be the type of plant, the type of soil, the mass of the soil, the depth of the soil, the light intensity, the volume of water added, the type of fertiliser, or even the time left at that temperature. Now there'll be loads more than what I've just spoke about, but these are the most obvious ones. One final important tip on this section is do not put height of plant as your constant variable. You're not controlling that variable, you're just measuring it. So now we're going to move on and look at the method. Now there's 2 formats for the method, and you'll need to check with your teacher which one they prefer. The first option on the left-hand side is the normal generic one, and that would be used for most science investigation write-ups. The one on the right is what's expected in the National 5 and higher biology assignments, so maybe something your teacher wants you to practice for these assessments. We'll start with the one on the left, so just using the general structure. So most normal science investigation reports would include firstly an equipment list, which is where you essentially bullet point a list of all the equipment and chemicals that you used in your experiment. Secondly, in this option, you would discuss safety over here. So you describe what was done to ensure participants in the experiment were safe. Really common example here would be wearing goggles to protect eyes from hazardous chemicals. Next, you would normally put in a labeled diagram, and this should be drawn as a line diagram using a ruler to make it really neat, and each piece of equipment should be clearly labelled. Finally, you would write a step-by-step account of your method. So sometimes it's best to write a draft of this and then add to it or edit it as you actually complete the experiment. Importantly, it should be in enough detail so that anyone could be given this and they could replicate the whole experiment exactly as you did it. So let's see this method section for an example experiment. So here you can see the 4 subsections I just discussed. So the equipment list with all the chemicals and the pieces of apparatus, including in this example measuring cylinders to measure the water, lamps, balances, etc. The label diagram over here details the temperatures and is really, really simple. The only thing I would say is missing from this example is I would have labelled the mass of the soil, the plant type, and the volume of water if these were kept the same in my experiment. For safety, there really isn't much to say for this experiment, so I've basically just added in a little bit about take care when using electrical equipment for this part here. So things like the balance, the water bath, etc. Put that in. For the step-by-step method, I've made it really detailed, so I'll read it out part by part, and you should see if you can visualise what to do if I gave you those instructions. So I've started with collect all equipment, and this will be the start of most of your methods. Because we've already listed the equipment up the top, we don't need to list it all again. Then I've said, weigh out 50 grams of soil using the balance and add it to each pot. So I said how I'd weigh it out here by saying I'd use the balance, and that's quite important. Add a plant to each pot, don't leave that step out. Measure out 100 mils of water using a measuring cylinder and add 100 mils to each pot. So again, I've mentioned the equipment I would use to measure, the measuring cylinder, and I've mentioned the exact volume of water and then what I did with that water as well. Measure the starting height of each plant using a ruler and record the results. Again, I'm pointing out that a ruler should be used to measure. Place each pot in a water bath at a different temperature, and I've listed the temperatures here as well. And I've said how I'm changing the temperature, so by putting them in the water bath. Leave for 48 hours, again detailing the exact time. And finally, record the height of the plant in each pot after 48 hours using a ruler and record the results. Again, saying how I'm going to measure it. Be careful, a lot of people miss this stage out. They say how they'll set the experiment up, but they don't always say how they would actually measure and get the results and record them. So as you can see, this is basically like a recipe. You're not saying I did this, you don't say I in your method at all, you're saying what to do, as if you're telling someone what to do and talking them through the stages. So now we're going to look at option 2 for writing a method. So this is specifically practicing how you'd write a method for the Nat 5 or higher biology assignment, so this bit on the right. Now, unlike the generic way to write a scientific method section, the assignment asks you to write a short summarised method, which would only be enough to let someone visualise the nature of the experiment and not actually carry it out. Therefore, you don't need to and shouldn't include specific volumes and concentrations. In contrast to option 1, this is also really, really short and should only be about 2 or 3 sentences long maximum. You should include these 4 points at the bottom, although the 4th one doesn't need to be included for every experiment. You should mention the independent variable, the dependent variable, how the dependent variable was measured, and any key chemicals used. So we're just now going to look at that in action. So I've colour-coded the 4 things we look for in an assignment method, but for this experiment we're only focusing on the first 3, as there's not a lot or any, to be fair, chemicals used in this particular example. So you can see how short this method is. I've said plants were placed in water baths at different temperatures, the height of the plants were measured at the start and again after 48 hours using a ruler. So I've mentioned the independent variable, which is highlighted in yellow, which is the changing temperatures. I've mentioned the dependent variable in blue, which is the height of the plants being measured, and I've mentioned how they were measured, so I've said using a ruler in green. So as you can see, these 2 options for the method are really, really different. You really need to check with your teacher which option they would like to use. If they haven't said specifically, it will usually be option 1, the generic method, but this one they might want you to do to get that practice in as well. So now we're going to move on to the results section, and we're going to focus on drawing a table first. So you should draw your table ideally before you complete an experiment and fill it in as you go, and this will make sure you don't make any mistakes, and also you'll collect all of your data properly. Your independent and dependent variables are really important again here and should be used in your headings copied exactly. So you must ensure you include units in your headings. They go at the top of the table in brackets beside the variables, here and here. They go at the top of the table in brackets beside the variables. They do not go beside your numbers in the table. You also need to plan whether you're going to repeat your experiment and make sure you enable enough columns for those. In this example, this is a generic table layout for an experiment that did 3 repeats for each independent variable. So 1, 2, 3. Now notice the independent variable is always on the left-hand column and the dependent's always going to be on the right. The dependent also goes over the top of multiple columns on the right-hand side, and this is because all of these columns will contain results for this dependent variable. This saves time and stops you having to write the dependent variable and units for every single one of these columns. There is space for 3 repeats for each of the independent variable conditions here, and also a column at the end for you to work out your average for each as well. So this is what it would look like in our example experiment. For the heading at the left-hand side, we have our independent variable, which is temperature, and we could fill this in before we did our experiment, as we know the temperatures that we were going to use in the experiment. We knew they'd be 10, 20, 30, and 40 degrees. Now, don't forget to put your units in the heading. They should be up here. In this case, degrees Celsius in brackets at the top, and not beside each number as you go down. We know that these ones are all in degrees Celsius because it's in the heading. On the right-hand columns, our heading is our dependent variable. So in this case, it's the increase in the height of the plant, and the unit is in centimetres. Again, I'm not putting centimetres next to every number. I'm just putting it in the heading. Now notice I've put increase in the height of plants. This would also be change in the height of plants as well, as we were actually measuring the start and end height of the plants and working out the difference. Those raw numbers are not included in this table, so this is the increase in the height of plants. This is not the height that they were at the start. There were 3 measurements taken for each temperature, and then an average was calculated in the final column. So on that note, just a quick note on calculations before we move on. So usually you would do some form of calculation in your report in biology, and usually this will be an average, as you'll most likely have conducted repeats for your experiment, just like in that one there. Now remember, you calculate an average by adding up the total number and dividing by the number of repeats. So in this case, for this experiment, I added in the first column for 10 degrees, 2, 3, and 2 together, and then I divided by the total number of repeats, which was 3, to get 2.3. Then I did the exact same for the repeats at 20 degrees, 30 degrees, and 40 degrees. However, there's another type of calculation in biology, which is really common, and that's percentage change. You'll need to know this for the exam, and it's often used in context of osmosis experiments. To do this, you work out the difference, divide it by the original, and then times by 100. If you feel you need to work more on this and you need to use this calculation for your report, go and watch the video on percentage change calculations before you do it. A final tip for this type of calculation is that you need to remember to include the plus or negative symbol in front of your final answer, depending on whether mass was gained or lost in the experiment. So now we're going to move on to look at drawing graphs. Now, the most important tips I can give for this are as follows. Firstly, do not mix up what should go on each axis. Your independent variable always goes on the x-axis, and your dependent variable always goes on the y. And again, this is why it's so important that you know your independent and dependent variable for your experiment. Secondly, don't forget to include your units in the axis labels. If it's temperature, you should have degrees C in brackets beside it, just like you would in the table. On that note, you should really get into the practice of using the exact same headings as the one that was in your table that you drew, or the one that's given to you in the table. Another tip on labels is that if you've repeated and taken an average of your results, you will most likely be plotting the average numbers, so you need to say this. On the y-axis, in your dependent variable heading, make sure you say average in front of your label. Always use a ruler and keep it neat, and in biology, remember to join the dots, don't draw a curve or a line of best fit. Finally, make sure your scales on each axis are even and consistent. Never jump. For example, don't ever go 1, 2, 3, 4, and then jump straight up to 8. It's best to always go up in 1s, 2s, 5s, or 10s. Never go up in 3s, for example, because it just gets complicated quickly if you're trying to plot numbers that are not divisible by 3. So if I were to plot the results of our example experiment, this is what it would look like. So this is the average results plotted. The x-axis has our independent variable, which is temperature, and also includes units in the label, so degrees C, and goes up in tens along the bottom, so 0, 10, 20, 30, 40. On the y-axis up here is our dependent variable, so the one we're measuring, and we're plotting the average results, so it reads average increase in height of the plant, and again we're using units, so centimetres is in brackets here. For this axis, it's best to go up in 2s, and this allows us to accurately plot the points in between. Your axis, both of them don't have to go up in the same intervals. You can have different intervals for each axis, that's fine. All points were then plotted accurately, really easy to see the dots, and you can't be more than half a small box out or you'll lose your mark. And finally, the points were joined using a ruler. So now we're going to move on and look at our conclusion. The conclusion firstly must answer the aim. That's really important. So at this stage, you need to go back and look at your aim and make sure you're answering it here. It also has to answer it using the results from your experiment. Additionally, you should usually explain whether this conclusion supported your hypothesis that you made before carrying out the experiment or not. If both sets of data are numerical, you will normally be able to see a trend and be able to describe that using the structure here. Now, basically, this is the structure I always follow when I'm writing a conclusion. So in conclusion, as the, and then insert your independent variable, increased or decreased, the, again, insert your dependent variable, increased or decreased. So you just write this, insert your independent and dependent variable, and choose between increased or decreased for each one. So we're going to look back at our graph very quickly, and we can see that there's clearly a trend. As the temperature increases along the bottom, so did the average increase in the height of plants. We can tell that because our line is increasing, it's going up. We can say in conclusion, as the temperature increased, the average change in the height of the plants also increased. We can also say that this proves my hypothesis from earlier that said that the higher the temperature, the taller the plants would grow. That was correct. So now what we're going to do is we're going to move on and look at the final section of the report, which is the evaluation. The evaluation should normally focus on 3 areas: the reliability of the experiment, the validity of the experiment, and the ways the experiment could be improved. For reliability, you should always ask yourself these questions. Did you repeat your experiment at each condition of the independent variable? So in our case, did we repeat it at each temperature? And did we take an average? For validity, ask yourself, did you control other variables that could have altered the results of the experiment? Did you only change one variable, the independent? And if so, you can list the variables you kept the same, and handily, you already have this list because you wrote it in your variable section near the start under constant variables. Finally, you need to think about how your experiment could be improved. I always suggest thinking about this question whilst you're completing the experiment, as you should always be reflecting on how you could make it better the next time, and it's easier to do that at the time rather than trying to remember back to you doing the experiment later. Improvements you can think of could be to do with reliability or validity, if you mentioned above that maybe you didn't repeat it or you didn't control some variables, or it could be on something completely different. So let's look at an example of an evaluation section from our example experiment. So for reliability, first of all, I've said this experiment was reliable because I did 3 repeats at each temperature and I took an average of my results. Next, validity. The experiment was valid because I only changed the temperature and kept other variables constant. These included the type of plant, the mass of soil, and the volume of water. Finally, I've identified one thing I could do to improve the experiment and explained why. So one factor that could have had an effect on my experiment was light intensity. Although I used a lamp, some plants were closer to the lamp than others, so some of them could have received a higher level of light intensity than the others. This could have had effect on my results, as those plants may have grown better because of the extra light and not the temperature. To improve this next time, I would use multiple lamps or would use a ruler to ensure all the plants were the same distance away from the light source. So not only have I mentioned what I did wrong, I've also then said why and what I would do next time to be able to improve it. So now we've been through all of the key sections that make up a science or biology lab report. I hope you found it useful and you now understand the basic sections and feel that you could hopefully apply them yourself now to any experiments you complete. When you're writing your next report, feel free to come back and listen to parts of this video again if there are areas you think you need a refresher on how to complete. Thanks for listening.